So welcome you all to another video lecture on cell biology. Today we'll study about nucleus. So let us start. From historical point of view, nucleus was first discovered by Robert Brown in case of your orchid plant. Now J. Hammerling first proved the hereditary role of nucleus in acetabulardia. Okay. Now depending upon the number of nuclei present in a cell, cells can be uninucleate, binucleate or multinucleate. Got it? If there is presence of a single nucleus, then we call the cell as uninucleate cells. If there are presence of two nuclei, then binucleate, and more than that we call as multinucleate cells. Now multinucleate cells can be sinusotic or syncytium. In case of sinusotic multinucleate cells, these are formed when free nuclear division takes place without cytokinesis. When cytokinesis is absent and when a nuclear division takes place, then that kind of multinuclear cells are known as sinusotic cells. But when the multinuclear cells are formed by the fusion of uninucleate cells, we call it as syncytium. Okay? When all the uninuclear cells fuse to form multinuclear cells, then we call it as syncytium. Got it? Now let us know about the structure of nucleus. As far as nuclear structure is concerned, nucleus consists of a nuclear membrane, a nucleolus, nucleoplasm and chromatin. So we will study one by one. First come to nuclear membrane. Nuclear membrane is the outermost layer that surrounds the nucleus. It is otherwise known as karyotheca. The presence of outer and inner membrane, that means the nuclear membrane is double membranous. Okay, there is presence of outer and inner membrane, and in between the outer and inner membrane there is presence of perinuclear space. So this space that is enclosed by outer and inner membrane is called perinuclear space. Now as far as the outer and inner membrane are concerned, they are around 90 to 100 angstrom thick. And as far as the space enclosed by outer and inner membrane is concerned, that is around 100 to 300 angstrom thick. Got it? Now outer membrane is continuous with endoplasmic reticulum. Okay, it is continuous with endoplasmic reticulum. Now nuclear membrane is not continuous. It is interrupted with many openings called as nuclear pores. Okay, it is interrupted with many openings called as nuclear pores, which maintain continuity between nucleocytoplasmic region. That means the nuclear membrane has got many pores which are called nuclear pores that maintain continuity between nucleus and cytoplasm. Okay, these nuclear pores are plugged with cylindrical proteins called as annulus. Okay, nuclear pores, they are not remaining open every time, rather they are plugged with cylindrical proteins called annulus. These annulus actually control the passage of ions and molecules into and out of nucleus. Okay, these annulus, which are actually cylindrical proteins, they control the passage of ions and molecules into and out of nucleus. Got it? So this is what your nuclear membrane is. Now come to the second one, that is nucleolus. It was first seen by Fontana in 1774 and the name was given by Bowman in 1840. Okay, it is a spherical, dense structure which is non-membranous. It does not have membrane and it is acidophilic in nature. Okay, acidophilic in nature. It occupies around 35% of the total volume of nucleus. Got it? The nucleolus is a spherical, dense, non-membranous structure which occupies around 35% of the total volume of the nucleus. And remember, it is acidophilic in nature. It remains attached to special type of chromosome called nucleolar organizing chromosome. It remains associated with special type of chromosome called nucleolar organizing chromosomes at particular sites of those chromosomes, at specific sites of those chromosomes, and those specific sites are called nucleolar organizing region or secondary constriction. You know about it if you go into the details of chromosome structure that I have already given you in this particular channel, you would be able to know what is secondary constriction. Okay. Now, nucleus consists of three regions: granular region, fibrillar region, then amorphous region. Granular region, fibrillar region, and third one is amorphous region. Got it? Now come to the nucleoplasm. It is actually the matrix of the nuclear membrane. Okay, it is actually matrix of the nuclear membrane. Otherwise, it is otherwise called as karyolymph or nuclear sap. Okay, it is very much denser. It is much more denser than the cytoplasm. Thus, it can easily be seen under the microscope. Very dense as compared to cytoplasm. It contains nucleolus and the chromatin threads. We have already studied about the nucleolus. Now we will study about the chromatin threads in the next slide. So these two structures lie immersed in the nucleoplasm. Okay, they contain basic or histone proteins and acidic or non-histone proteins. These contain basic or histone proteins and acidic or non-histone proteins. Actually, these nucleoplasm, this is the matrix. This nucleoplasm is the matrix in which nucleic acids, your histone proteins, non-histone proteins, everything else lie. Got it? So now we have already studied about the nuclear membrane, nucleolus and nucleoplasm. Then we will go to the last one, that is chromatin threads. Okay, you know about chromatin threads. These are nothing but thread-like structures made up of DNA with histone proteins. Okay, these are thread-like structures formed by the association of DNA with histone proteins. There is not much of difference between chromatins and chromosomes. They are chemically similar. Only their degree of packaging, they differ in their degree of packaging. That I have told you in the chromosome part. Okay, I have in the video that I have uploaded in this channel on chromosome structure, that I have told you what is chromatin and what is chromosome. Just same thing, only they differ in their degree of packaging. The relaxed state is the chromatin. Okay, the more condensed coiled one is the chromosome. Okay, so the chromatins are thread-like structures formed by the association of DNA with histone proteins. These are nothing but relaxed state of the chromosome found in the interphase nucleus. Got it? So this is what the structure of nucleus. So what we have studied till now, as far as the nucleus is concerned, it consists of nuclear membrane, nucleolus, nucleoplasm and chromatin threads. Nuclear membrane is the outermost layer that surrounds the nucleus, consisting of outer and inner membrane. It is otherwise known as karyotheca. The nuclear membrane encloses, both the outer and inner membrane. In between them, there is presence of perinuclear space. Got it? The outer membrane is continuous with the endoplasmic reticulum, whereas the inner membrane, outer membrane is continuous with endoplasmic reticulum. Inner membrane actually encloses the matrix of the nucleus, that is your nucleoplasm. Got it? The nuclear membrane gets interrupted with small openings. That is called, those are called your nuclear pores. Okay, these nuclear pores remain plugged with cylindrical proteins called annulus, which actually control the passage of molecules or ions between the nucleus and the cytoplasm. Got it? Now, nucleolus, we studied about nucleolus. It's a spherical dense structure, non-membranous structure, which occupies around 35% total volume of the nucleus. It is associated with special type of chromosome called nucleolar organizing chromosome, which at specific sites called as nucleolar organizing region or secondary constriction. Then we studied about nucleoplasm, the matrix enclosed by nuclear membrane, where everything else of the nucleus lie immersed. Okay, your histone proteins, non-histone proteins, all the nucleic acids, nucleolus, all of them lie immersed in nucleoplasm. Got it? Then last one was the chromatin threads, which are nothing but thread-like structure formed by the association of DNA with histone proteins, which are similar, chemically similar to those of chromosomes. Got it? So now let us study about the functions of nucleus. It controls all the genetic and metabolic activities of the cell. It acts as a genetic repository of cell. As far as the nucleus is concerned, all the genetic and metabolic activities of the cells are controlled by nucleus and acts as a genetic repository of the cell because they contain chromatin threads or chromosomes. As they contain chromatin threads, obviously all the genetic informations are stored within them. Got it? Similarly, the nuclear loss found inside the nucleus participates in the assembly of ribosome during your protein synthesis. Got it? So, this is what this functions of nucleus. Okay. And then we will go to the diagram of nucleus. This one is the diagram of nucleus. As you can see, there is presence of outer and inner membrane. Just a minute, let me draw here. So, as you can see, there is presence of outer and inner membrane. This one is the outer membrane, this one is the inner membrane. Got it? So, outer membrane remains associated with endoplasmic reticulum, as you can see here. Outer membrane remains associated with endoplasmic reticulum. So, there in the nuclear membrane, there is presence of pores called nuclear pores. This is the nuclear pores. Nuclear pores. Got it? The nuclear membrane encloses your nucleoplasm. Nucleoplasm. This one is the nucleoplasm in which nucleolus, then chromatin threads, they remain immersed. Got it? So, another one that I have not shown here, probably no, I haven't shown here, that is in between the inner and outer membrane there is presence of what? Your perinuclear space. Okay. Then you can see here that these rounded reddish structures, these are ribosomes associated with endoplasmic reticulum. Okay. I hope this particular video will be helpful for you in understanding the structure of nucleus. Thank you very much.